our next speaker is Nguyen. Ali got her PhD at Rutgers in molecular biosciences and is currently doing her postdoc at the Whitehead Institute.
Thank you. Thank you for that introduction. As Jen said, my name is Ali Nguyen, and I'm a postdoc at the Whitehead Institute in the Cheeseman Lab. And I'm really excited to be here today to tell you about my work, which really seeks to understand how cells might tailor core cellular processes such as cell division and genome organization, processes that we generally think of as invariable, how they tailor them to fit their particular needs. And so I got really interested in this as a graduate student, actually, where I was working on what I think is a pretty clear example of when we need to tailor cell division. And that's when our goal is either to generate a diploid clonal cell, or if we want to make a haploid gamete. And so how do we change cell division? And so for my postdoc, I was really interested in thinking about, well, what are other situations in which a cell might need to change how it does cell division or genome organization? What about in somatic tissues? Can we identify differences? And so if we think about what might influence gene requirements, I think some obvious examples are cell type. The needs, especially when we think about genomic organization, between a retinal cell or a fibroblast are going to be incredibly different. The state that that cell is in, is it cycling, or is it entered into a quiescent or even a senescent-like state? The disease state of that cell, because we know that with extreme aneuploidies or other mutational burdens, this creates really unique challenges that cells have to overcome in order to continue to proliferate and survive. And then finally, if we can imagine all these differences existing within cells of a single organism, it's not a far stretch to believe that we'd find differences across even closely related species. And so for my short time with you today, I want to really highlight just 3 key areas that my research focuses on that really uses differential gene requirements as a way to better understand cell division and how we can use it to define difficult to study proteins, how we can use it to discover new protein players in chromosome biology, and then finally, some of my more recent work, which really seeks to understand how gene requirements change across organisms. And so in thinking back about my cell division core, I was really interested for my postdoctoral work of thinking about how might cell division change, but across mitoses. And so I joined the Cheeseman Lab, and in the Cheeseman Lab, we think a lot about many aspects of cell division, but one part in particular we like to focus on is how chromosomes physically attach to spindle microtubules so that they can be segregated. And I'm showing you a microscopy image here where the DNA is shown in blue and the spindle microtubules are shown in green. And that point of connection is known as the kinetochore. And this is actually a large assembly of proteins that must precisely assemble to achieve this function. And I don't want you to remember any of the names of these proteins so much that I hope you can appreciate there's a lot going on. And because this function is critical to cell proliferation, we'd imagine that any of the proteins that are part of this larger complex should be universally required. But this is not actually the case. And so we can ask this question now with the advent of CRISPR-Cas9 and large-scale screening approaches and say, okay, of any of the proteins that are part of this complex, are they universally required? And one of my favorite resources for this is the cancer dependency map. This is out of the Broad Institute, where they've now done genome-wide CRISPR-Cas9 screens across over 1,000 different human cell types. And so if we just look at any of the proteins in here, for example, NDC80, it was required in 1,095 of the 1,095 cell lines that it was knocked out in. Similarly, if we look at SEMP-N or SEMP-K, we see the same thing. These are genes that are universally essential, as we'd predict. But there's another complex here, the SEMP-O complex, that behaves very differently. Here, only in 94 of the 1,095 cell lines that it was knocked out in did it actually impact cell proliferation and survival, something listed as strongly selected. And so I was really interested when I started my postdoc in figuring out, well, what is this protein complex doing? It's actually really strongly conserved from yeast to humans. It's always expressed in cells. Why are they making it if they don't seem to need it? And so what had made understanding the function of the SEMP-O complex really challenging was this differential requirement. Because when we just used our standard cell lines in the lab, such as HeLa, and we knocked it out, nothing happened. This is a SEMP-O knockout that I generated, and this is a perfectly happy mitotic cell. And so my approach to tackling this question was to go back to that cancer dependency map data, identify a cell line that is sensitive to the loss of the SEMP-O complex, and then use that as a better system to study what this protein complex is doing. And so now you can see we have a really robust phenotype that we can follow up on. But importantly, what I'm really interested too is going back and comparing this to a situation where it's not required and using this to try to tease apart what's driving this differential requirement. And so for the sake of time, I'm going to sum up a lot of work in a simple graphic. And what we found was that the SEMP-O complex is really important to recruit this kinase that regulates mitosis, PLK1. And this is a universally essential gene. And so you might ask yourself, how does it recruit a universally essential gene if it in itself is not universally essential? And what we found is that the recruitment of PLK1 is actually done by two different arms. It's done by the SEMPO complex, as well as another protein, BUB1, that together make sure that a sufficient amount of PLK1 is recruited to kinetochores. And so normally, when you get rid of the SEMPO complex, you still have this other job. It's able to recruit enough PLK1. But what we hypothesized is maybe there are situations where BUB1 is perturbed, and that's creating a sensitization to the loss of the SEMPO complex. And this is exactly what we see. So if we screen across cell lines that are sensitive and not sensitive to the loss of the SEMPO complex, we could always see that in those cell lines that are sensitive to the loss of this complex, they always seem to have less BUB1 localized to kinetochores. And so we hypothesized that here in this situation, you're not able to recruit enough PLK1 to do its job. It's above a certain threshold. And so if this model is true, then we should be able to create the situation really in any cell type. We should be able to titrate away the amount of BUB1 to cells and make them sensitive to the loss of SEMPO complex, and this is exactly what we can do. And alternatively, we can rescue cells that are sensitive to the loss of the SEMPO complex simply by just giving them more BUB1. And so using this approach, it was really great to finally be able to entangle what is the function of the SEMPO complex and precisely actually how PLK1 is localized to kinetochores, two important questions that had sat around for a long time in cell biology. But I think even if we think about this bigger, it's a great opportunity to really identify differences between healthy cells and disease cells, situations that we can use to act as an Achilles heel for cancer and develop more targeted therapeutics while leaving your healthy cells intact. And so I told you how I'm using differential gene requirements to study known genes, but I want to show you how we can also use differential gene requirements to identify new protein players in your favorite pathways, and for me, that's chromosome biology. And so in addition to thinking about how cells segregate their genomes, I'm also really interested in thinking about how they organize them. And a major component to DNA organization is the cohesin complex. And this is a protein assembly that forms a ring-like structure that helps to organize your DNA into loops. And the ability for the cohesin complex to do this is dependent on a variety of cofactor molecules that dictate where cohesin goes, the stability of it when it unloads. And we also know that the regulation of this is going to be wildly different across cell types, even within a single organism. Because again, how you organize your DNA to regulate, for example, transcription is going to be different for the needs across cell types. But we actually have a really poor understanding for how cohesin functions and what these different cofactor molecules are. And so what I want to do is to identify these cofactor molecules to go back to those large databases like the cancer dependency map. But instead of saying, okay, I want to study the SEMPO complex, let me use this to identify these cell lines are going to be most sensitive. Now I can look at the general behavior of these proteins across many, many situations. And so for example, if I use a known cohesin protein as a bait, I can then ask, are there any other genes or proteins that behave very similarly across all these situations? And because similar trends predict similar functions, we can then assume that this is also going to be in this pathway. And so using this approach, I've identified multiple proteins that I predict are involved in cohesin regulation. And for the sake of time, I'm going to focus on only one of them today, PRR12, very briefly. And what's really cool about this approach is not only does it help identify proteins to follow up on, but it also gives you a testable hypothesis based on who these proteins are co-coordinating with. And so importantly, PRR12 actually comes out as codependent with 3 different proteins that are involved in cohesin regulation, NIPBL and MALT2, and STAG1. And importantly, these are all proteins that confer stability to the cohesin complex. And so from this, we can make the hypothesis that maybe PRR12 also contributes to cohesin stability. And so at heart, I'm a microscopist, and so I wanted to answer this question using that approach. And so what I wanted to do is generate a cell line that I can knock out PRR12 in. I use an inducible knockout system. But okay, I want to study cohesin. We know cohesin is really important to cells. It's going to be really hard if loss of this protein does confer a problem to cohesin and the cells are just going to die. And so to get around this, I built the system in an NIH3T3 background because I can induce these to go into a quiescent-like state. And that way I can look at genomic rearrangements or changes while removing secondary defects from, that will cause cell death. And so using this approach, if I just look in the control cell using an antibody against SMC1, a core component of the cohesin complex, I can see some level of cohesin localized in the nucleus, and that's just quantified over here. And then as a control, I can knock out the core cohesin complex itself, and I can show that cohesin is lost, but the cells are able to survive. And so what happens when I knock out PR12? And to our excitement, we see a significant decrease in the localization of cohesin in these cells. And so I don't have time to show you more of the story today, but to simply say that we're really excited about following up on what is the specific mechanisms by which PR12 is regulating cohesin, and I have some exciting data to suggest it's through a direct interaction with the complex. But I think even more broadly, I'm really excited about continuing to use this approach to identify new players in my favorite pathways, but also what you might be interested for your pathways. And so using this to identify more chromatin remodeling proteins, but also new players in cell division, because we still don't know what a fifth of our proteome does. And so this is a really great way to start assigning potential functions. And so using this, once I identify those proteins to follow up on, my lab will ask, what is the function of these proteins? Why is this function situation specific? And importantly, what's driving this diversity? And so for the last couple minutes, I want to take a step back and ask, okay, I've looked at all of these differences that might exist across cell types within a single organism, but what about across organisms? We're often using different organisms as a proxy for human health, but we fail to appreciate the differences that exist. And so when I started thinking about this, I was actually really surprised that there hasn't been a lot of CRISPR-Cas9 screens performed in mammalian species outside of humans. There's a few select studies that have been done in mouse, and I'm showing you one such example where I've just plotted the CRISPR screen score from mouse cells and then a comparable human cell line. And so on this plot, every single dot represents a single gene, and the score just represents how much loss of that gene impacted the ability of those cells to proliferate and survive. And so a more negative score means it was worse for the cell. And so I want you to take away a couple things from this. First, we should appreciate there's a lot of similarity. I'm not trying to say that these aren't good models for human health. But there's also some areas that I'm really fascinated by. And that's these two areas that I'm pointing out. In the top section here, these are genes that are essential in mouse, but not human. And then also here, genes that are essential in, this was genes essential in human and not mouse. And then over here, genes essential in mouse and not human. And there's a lot of genes here to follow up on. And importantly, two of them that I've talked to you about today, SEMP-O comes out. So I told you that this protein is actually not required for most human cell types, but we know it's embryonic lethal in mouse. And so trying to figure out what's driving that difference. And then another protein I talked to you about today, PR12, also has some really interesting differences between human and mouse. But it's not just how these proteins behave in the normal context, but also how these pathways change when the cells are challenged in some way. And so this is an assay we use a lot in the cells where we treat cells with a mitotic inhibitor, STLC. This is going to collapse the mitotic spindle and cause a really robust arrest in mitosis. And so this is just cells under a live imager, human cells here on the left, and cells from a rat on the right. And in human cells, when they're treated with this inhibitor, they all round up in mitosis and they get stuck there. And we can just quantify that here. But when you start screening this across species, and this is work done with a really great rotational student I had, we can see that they behave really differently. If you look at these rat cells, they escape mitosis pretty quickly, and they enter into a G0-like state, escaping from mitosis. And so I'm really interested in following up what's going on here. And so going back to say that there's not a lot of great data sets that we can start scoring through across these organisms. What I'm doing is generating CRISPR-Cas9-based screens across a variety of mammalian species, importantly using a similar cell type across these different situations. And then we can also not just ask what happens when these genes are knocked out in the normal situation, but also what happens when they're challenged with a variety of mitotic inhibitors. And then this is just to simply quickly say, I've started doing these screens. I have the first data from my rat screen. And there's a lot of really interesting pathways that have already started coming out. Core components of that kinetochore complex that I told you in the beginning are essential do not show that same essentiality in mouse, and then a variety of other things that we're following up on. And so I've told you a lot how I'm using differential gene requirements to study cell division and genome organization. And I'm taking all of this with me to start my own lab when I go on the market. And so the Nguyen lab will use differential gene requirements to understand, discover, and explore chromatin biology, and to ask questions such as, how does cell division change across really diverse situations? How does the genome organization change to facilitate the needs of specific cells? And importantly, how can we identify new players in our favorite pathways? And so I think not only is this important to understand core molecular processes in cell biology, but also just to appreciate the really beautiful diversity that exists across cells. And so with that, of course, I want to thank those who have really been instrumental in my work now and in my future. So my lab and my advisor, and then of course all the variety of core facilities at the Whitehead that have been really instrumental in designing these. These animals do not have libraries that we can pull to generate these guides, so there's a lot of work that goes into it, and these cores have been really fundamental to that. And then of course funding and all of you.
Should I?
Hi, that was Brittany from Georgia Tech. That's a really cool talk. I was really surprised that you have so many genes that are uniquely essential across like different species. I'm curious, like if there's like other orthologs in like a mouse that makes up for the function of that gene.
Yeah, absolutely. So that's one of the things we're finding from our first screen. Even when we were designing the guides, I mean, you'd be surprised how many, once you start getting outside of like primates, instead of being one-to-one, it's one-to-many, right, when you're designing these guides. And so we designed, anytime there was that situation, I actually designed guides individually to all of those orthologs. And we do 5 guides per, so each ortholog got 5 independent ones. And so when we separate them, we see some really interesting differences in the behavior. But that's what drives, for example, evolution of protein function, right, is when you can have one still doing the core job, and now these orthologs or other paralogs, I always forget, homologs, I should know this, that change along with the core function that's going on. And so that's something we're also keeping in mind. We absolutely see that.
Yes. So yeah, beautiful talk. So at the beginning of the talk, you show some experiments with the knockout of SEMPIO and how different cells have different sensitivities to the lack of it. So do you see any trend in the cells that are more susceptible to the lack of SEMPIO? For example, cells that may tend more to have chromosomal abnormalities or, yeah.
You know, that's a good question. Most of it that we found were sensitive to loss of the SEMP-O complex were actually blood cell types and leukemias. So chronic myeloid leukemias. So I haven't looked more into what might be driving aside from BUB1 changes. And I haven't fully gotten into what's driving the BUB1 change. And that's something I'm really interested in following up. And then in thinking about the differences that we see across mouse, is it the same thing that's explaining that? Or are there different roles between development and more adult cells? Because a lot of those blood cell lines are more closer to like the stem lineage than maybe a HeLa cell, right? And so there could be interesting differences there that we have yet to untangle.
Thank you.
So I think you might have already mentioned this. What is the reason behind K562s or other cell lines not having enough BUB1 to begin with?
We don't know. I know they have less total protein, but I haven't looked at, and I couldn't identify any noticeable mutations or things like that that would explain what's going on. So whether there's something in the scaffold further in that doesn't allow it to localize and the protein levels being reduced, could be that it's being degraded because it can't do its job. I haven't looked into it, and I think it's an important question to start thinking about, especially if we want to use this as a therapeutic target, right? Now this creates a really cool pathway that you should be able to titrate, say, BUB1 levels or PLK1 levels. PLK1 is a target already in cancer therapeutics and in myeloid or chronic myeloid malignancies. They've shown that it actually they're more sensitive to PLK1, and they didn't know why. But I think we need to follow up more what's happening to BUB1 itself.
Yeah. So I was just curious about, it's maybe related to Brittany's question, but like in that graph where you showed the genes that are essential in human but not mouse, and then in mouse but not human, it looked like there was like a handful that were human but not mouse, but then like a ton that were in the other quadrant or whatever. And I don't know if you just, what are your thoughts on that?
Yeah, so there is a general downshift that mouse cells seem to be more sensitive. Some of that could be that we're comparing mouse cells that are usually, there's not a lot of great comparisons out there. And so a lot of times we're comparing like cancer cells that kind of are good at just getting away with a lot of problems versus more normal cell types. It's also hard, so this is not my data. And CRISPR scores are very arbitrary. We can learn things from this, but I don't want to compare this as a finite. A lot of this shift could just be poor comparison. And so that's why I wanted to do these screens and really control the cell type. I wanted to control how I'm calculating that CRISPR score. So now people move to like a Z-score where you start taking into changes across the broader system and how just cells or how proteins behaved in general and try to adjust things for that. That hasn't been done here. So part of it could be just simply you're comparing a little bit apples to oranges here. I do think, because I followed up on all of these proteins, there is really interesting differences, but you have to take that caveat into account of, which one of these are real follow-ups and which are...
And kind of like what you said before about just like, is it actually a true homologue? Like, are you comparing?
Yeah.
Yeah, right.
Thank you. Great. Thank you, Dr. Nguyen.